I'd like to introduce our team. The PI is Professor Dr. Nishimura, who is an AAS fellow. He has participated in molecular and nanotechnology. He apologized that he can't be here today because he was invited as keynote speaker in Japan, but he's online now. So Dr. Hokugo is co-PI and he is assistant professor of UCLA plastic surgery. He has been involved in regenerative medicine and wound healing studies. And I'm Dr. Hiroko Okawa. I'm working as project scientist in Dr. Nishimura's lab. I have been involved in tissue engineering and high-throughput screening research. Dr. Jari is a plastic surgeon as a consultant, our team. Our main subject is shown in this slide. Despite much effort, effective therapeutics to minimize wound scarring are not yet fully developed. To improve wound healing and minimize scarring, our solution is to use small molecule inhibitor of neuronal path domain 2, commonly called Npas2. We have a competitive edge for obtaining novel target and preclinical safety profile and cost saving. For the next step, I'd like to discuss the problem we are trying to solve. Worldwide, 100 million patients develop scars from elective and trauma operations alone each year. In 40 to 70 of surgical cases, patients experience hypertrophic scarring. Particularly in facial area, hypertrophic scarring can be psychosocial challenging. Hypertrophic scarring is characterized by the excessive amount of collagen and raised scar. This is typical histology of hypertrophic scar. You can see the dense collagen fibers strongly stained by blue. Current standard treatment for hypertrophic scarring include steroid injection and others. Next, I'd like to share an interesting relationship between circadian rhythm and wound healing. The One England group reported in Science Translational Medicine in 2017 that human burn wounds that occurred during nighttime took much more time to heal than those that occurred during daytime. They also did mechanistic investigation to see why daytime is better. Wound healing requires fibroblast migration. To test fibroblast migration, they used in vitro scratch model that is common method for in vitro wound healing. They cultured skin fibroblasts on the plate and they just scratched the plate at nighttime or daytime. As you can see, fibroblasts scratched at daytime migrate faster than fibroblasts at nighttime. This result shows faster migration means better healing. Many clock genes have been reported to date. We have to define some target genes. Fortunately, we discovered one target gene, Npas2. In other projects, our team is working on the investigation of the dental implant. We surgically placed small titanium implant on rat femur. After 4 weeks, we performed 4 genome microarray of peri-implant tissue. Interestingly, Npas2 is the most important in the role of wound healing with implant. We generated Npas2 knockout mutant mice, and did the same implant surgery as before. In the wild type group, dense collagen deposition. In the wild type group, dense collagen tissue around the implant is beneficial for bone integration. Surprisingly, Npas2 knockout mice did not form dense collagen fibrous tissues. So we realized that dense collagen fiber is the common structure of hypertrophic scarring. Thus, we hypothesized that the suppression of Npas2 decreased fibrosis formation. We tested this hypothesis in skin punch wound healing model using wild-type mice and Npas2 knockout mice. As you can see, Npas2 knockout mice improved wound healing and minimized scarring. We investigated the effect of Npas2 suppression on skin fibroblasts in vitro. We performed scratch wound healing and collagen gel contraction assay known as in vitro wound healing model. Npas2 knockout fibroblasts shows high migration and contraction ability. These results suggest that Npas2 knockout skin fibroblasts improve wound healing in vitro model. Taken together, Npas2 is a novel chromotherapeutic target since Npas2 suppressor has high efficacy to accelerate wound healing. And also, Npas2 knockout mice did not show embryonic or developmental pathology in mice. Therapeutic suppression of Npas2 expected to cause minimum side effect. Npas2 would be a safer target than the alternatives. Next, I would like to show our technology platform to find Npas2 suppressive compound. First, we created mouse skin fibroblasts with reporter genes and started screening over 1,000 FDA-approved compounds. After screening, we got 10 hit compounds that downregulate Npas2. However, some hit compounds downregulate Npas2 because of their toxicity. So we combined cell viability assay with HTS and succeeded to eliminate 2 false positive and obtained top 5 hit compound. So one of the hit compound named DAN1 was fully evaluated IC50 and LC50. We tested the in vitro wound healing ability of DAN1 using previously described method. These results was that DAN1 accelerated fibroblast migration and gel contraction. To test in vivo wound healing using DAN1, we created 1.5 by 10 mm skin split model with suture in the middle and designed 3 groups. When we only sutured skin, visible wound healing was not good. The vehicle control with 10% dimsol looks better, visible wound healing was looked better. This is understandable. The moisture vehicle may help wound healing. In addition, DAN1 in 10% dimsol showed the best healing. We performed mason trichrome staining, which stained collagen deposition as blue. We found thick blue collagen deposition not only in granulation tissue but also in the peripheral tissue. The vehicle control with 10% DIMSO was similar to control. The thick collagen deposition was observed. By contrast, down one in 10% DIMSO showed very small area of collagen deposition. These results indicated that down one improved split wound healing with minimal scarring. We looked at therapeutics in clinical development and found that current approaches are focused on fetal-like wound healing. However, this strategy of fetal-like wound healing has produced only mixed results to date. These companies have halted development of their assets. Our therapy is completely novel. We have an innovative target that modulates the circadian rhythm to promote scarless wound healing. I'd like to show market prioritization. High patient incentive to adopt is aesthetically focused patient. Particularly, our technology platform is targeting for these cases. High surgeon incentive to adopt is for financial incentive to maximize aesthetic outcomes. In addition, our target compound improves scar appearance, combining with the surgical techniques designed to minimize scarring. Regarding patent, we have already submitted provisional patent applications for our invention. Regarding safety and efficacy of small molecule compound, we are going to optimize HIT compound to evaluate maximum potency without toxicity to apply lesion of skin. About licensing issue, we plan to test analog compound for possible new chemical entity. For translational potential of Npas2 inhibitor in phase 1 study, determining an in vivo wound model will reflect similar outcomes during clinical development. This is our current timeline and milestones. Milestone 1 and 2 have been completed with HTS, and at least one HIT compound was discovered. We propose to complete biological characterization of all HIT compounds and their analogs using human fibroblast in milestone 3. Milestone 4 will determine the formulation of the candidate compound. Then we will establish the therapeutic efficacy using in vivo mice wound healing model for future FDA application. This is the end of my presentation. In summary, we would like to improve wound scarring by regulation of circadian clock. Our technology is small molecule inhibitor of Npas2. We have successfully developed our technology platform and discovered HIT compound. Thank you very much.